So it looks like 16.7 will be easier than 16.5 and 16.4, at least shorter. I'll start by reading the problem. The locations of numerous LAC-I- and LAC-I-S mutations have been determined with the DNA sequence of the LAC-I gene. Among these, LAC-I mutations were found to occur in the 5' upstream region of the gene, while LAC-I-S mutations were found to occur farther downstream in the gene. Are the locations of the two types of mutations within the gene consistent with what is known about the function of the repressor that is the product of the LAC-I gene? Okay, so I think this is pretty simple. So what the question is saying is if this is a diagram of the LAC-I gene, which encodes the LAC-I repressor. So LAC-I gene. Now, this 5' region here is where the mutations for what? For LAC-I minus mutations are found there, and down here these are the mutations that lead to LAC-IS, so the super repressor. And we remember that this is how we like to diagram the shape of the LAC-I protein, and this part up here is the operator binding site. This one down here is the lactose binding site. So operator binding, lactose binding down here. And okay, so this makes sense, right? So we have two different parts of the protein. The LAC-I mutations lead to problems up here, where we get rid of that binding site here. Okay, so it makes sense that they would be in a different part of the gene than, say, mutations down here, which eliminate the lactose binding site. Okay, which would be found in a different part of the gene. So this is completely consistent with what we understand, with what we know about how the LAC-I minus mutation works, eliminating the operator binding site, and the LAC-I super suppressor mutations work, eliminating the lactose binding site. Both those binding sites are in different parts of the protein, so it would make sense that the mutations for each are found in different parts of the gene.